Pull up a tomato plant at the end of August after a full season of growth, and the root ball tells you everything. Not the fruit, not the leaves, the root ball. In a garden where one simple kitchen ingredient was applied 3 times across the season, that root ball is not recognizable as belonging to the same species as the plant growing 6 feet away without it. The mass is 2, sometimes 3 times denser. The lateral branching runs in every direction, like a map of river tributaries. The fine root hairs are so numerous they hold the surrounding soil in a tight, dark fist that barely crumbles when you lift it. The plant growing without the ingredient next to it slides out of the ground clean, a thin, pale taproot with a few stringy branches hanging off it like wet thread. Same variety, same soil, same season. 2 completely different plants underground.
And the ingredient that made the difference cost less than $1, ships in a small paper packet, and sits in the back of nearly every kitchen pantry in the United States without anyone giving it a second thought. This is not a gardening rumor passed between neighbors over a fence. The effect on root biomass has been measured in peer-reviewed research published in Crop Science in 2014, in the journal Plants in 2023, and in Frontiers in Industrial Microbiology in 2024. The scientists who ran those studies were not looking for a garden hack. They were studying fundamental plant biology. And what they found underneath the soil completely rewrote a textbook assumption that had stood unchallenged for more than a century. The ingredient is plain dry baker's yeast, 1 standard packet. The kind sold in every grocery store in America for somewhere between 60 cents and $1. Saccharomyces cerevisiae, the same single-celled fungus that has made bread rise in American kitchens since before the country had a name. And the reason you have never seen it on the shelf at a garden center, sitting next to the rooting powders and the bloom boosters, is not a conspiracy, not a trade secret, and not because it does not work. The reason is pure physics, and once you understand it, the entire story of why gardeners over 55 are the ones who tend to know about this, while younger gardeners keep paying $12 for a small tub of synthetic powder, starts to make a very specific kind of sense. But before the recipe, before the research numbers, before the physics of why a store cannot sell this to you even if it wanted to, there is a more important story to understand. The story of what actually happens underneath your garden in the dark, and why almost every product sold in the gardening aisle of a major retail chain is designed to address the wrong end of the plant entirely. Most gardeners spend their entire lives looking up at the canopy, up at the blossoms, up at the swelling fruit. When a pepper plant looks pale and tired in July, the first instinct is to spray something on the leaves. When a tomato refuses to set fruit, the first thought is to scatter granular fertilizer across the soil surface and water it in. When a freshly transplanted seedling wilts on the first hot afternoon, the instinct is to give it more water and shade it with a piece of burlap and wait. These responses are not wrong exactly, but they are addressing the symptom while the actual problem is happening 12 inches underground in a place no one ever looks. The root system of a vegetable plant is not a passive structure. It is not a drinking straw. It is the entire engine of what happens above the soil. Every nutrient that swells a tomato to full size, every molecule of nitrogen that deepens the color of a pepper, every drop of water that keeps a squash vine from collapsing under a heat wave in late July, all of it must first pass through the root system. If the roots are thin and shallow, the plant above is running a powerful machine on a fuel line the width of a garden hose. If the roots are dense, deep, and branching in every direction, that same machine runs on a pipeline. The harvest you see in September was decided in April and May by what happened underground during the first weeks after transplant. The garden center aisle was not built around this reality. It was built around what is visible, what is colorful, what can be bottled and labeled and stocked on a shelf for 6 months without spoiling. Leaf sprays, bloom boosters, fruit setters.
Foliar feeds with bright labels showing enormous red tomatoes. These products exist because they are stable, profitable, and easy to ship to a warehouse in Texas in July without anything bursting, leaking, or dying on the way. The root zone is harder to sell to. You cannot see it. You cannot easily photograph it for a package. And the most powerful thing you can do for it happens to be alive, unstable, and essentially impossible to bottle at commercial scale. That last fact is the entire reason this video exists. There is a living organism that, when introduced to the root zone of a vegetable plant at 3 specific moments in the growing season, triggers a hormonal cascade, delivers a wave of pre-digested minerals directly to the root surface, and physically colonizes the soil around the roots in a way that protects them from certain fungal pathogens. It costs less than $1 per application. It can be prepared in any kitchen in 30 minutes using equipment that every home already owns. And it cannot be sold on a retail shelf because the moment it is sealed into a bottle and shipped to a warehouse, the living organism inside begins generating carbon dioxide gas at a rate that will cause the container to bulge, crack, and fail within 48 hours at room temperature. There is no preservative that can stop this without also killing the organism, and a dead version of this product produces almost none of the effects that make it worth using. So the recipe has lived where it has always lived, passed between gardeners who learned it from gardeners before them, written in the margins of seed catalogs, mentioned in letters between rural families during the middle decades of the last century, when every household that kept a kitchen garden also kept a standing jar of dry yeast on the pantry shelf as a matter of basic domestic routine. The science that explains why it works did not arrive until 2010. The observation that something remarkable was happening in the root zone when yeast was present had been around for generations before anyone put it under a microscope. What that microscope revealed in 2010 at a university in Australia is the part of this story that genuinely surprised plant scientists. And it is the part that changes the way a gardener sees every plant in the garden from the moment they understand it. Because it turns out that a tomato root does not simply absorb nutrients dissolved in water. It hunts. It reaches out, identifies a living microbial cell in the surrounding soil, and pulls that cell physically inside its own tissue. Then it strips the cell apart and extracts the contents directly. Nitrogen, phosphorus, stored proteins, trace minerals, everything the microbial cell had accumulated while feeding in the surrounding earth. The root does not wait for nutrients to dissolve and drift toward it. It goes and takes them. That process has a name now. It was named by the researchers who first observed it, and it appears in dozens of follow-up studies across multiple plant species published over the 14 years since the original observation. Understanding it is what separates a gardener who uses yeast water as a vague folk remedy from one who uses it as a precision tool, applied at the exact 3 moments in the growing season when it produces the largest measurable return. The name, the mechanism, and the recipe are all coming. But the root is where the story starts, and it is the place almost every piece of gardening advice sold in America completely ignores. In the summer of 2010, a research team at the University of Queensland in Australia loaded a tray of young tomato seedlings into a laboratory fitted with a confocal laser scanning microscope. This is not the kind of microscope you find in a high school biology classroom. A confocal system fires a focused laser through a living tissue sample and assembles a 3-dimensional fluorescent image in real time, allowing a researcher to watch biological processes unfold inside a living cell without cutting the tissue open. The team, led by Dr. Chanyarat Lonyen, had introduced living Saccharomyces cerevisiae cells, plain baker's yeast stained with a fluorescent marker, to the root zone of the seedlings. Then they watched what happened next. What they saw had not been described in any plant science literature before that moment. The root tip cells were not passively absorbing nutrients from the surrounding solution. They were reaching out, making physical contact with the yeast cells floating in the water around them, and then pulling those cells inside. Not dissolved minerals, not free-floating amino acids. The entire living cell, intact, was drawn through the root cell wall and into the space between the wall and the membrane, a region called the apoplast. Once inside, the plant cell produced a controlled burst of superoxide, a mild oxidative signal that broke down the yeast cell wall from the inside and made the internal contents of the fungus available for direct absorption. Every nutrient the yeast had accumulated while feeding on sugar in the warm water, nitrogen compounds, phosphorus, stored proteins, B vitamins, trace minerals, was now available directly to the root cell that had swallowed it. Then the observation became genuinely strange. Some yeast cells survived this process. The plant cell dismantled the wall but did not destroy the organism entirely. The surviving yeast cells were expelled back out through the growing root hairs into the surrounding soil. Once back in the earth, they rebuilt their cell walls, gathered new nutrients from the surrounding environment, multiplied, and were eventually consumed again by the roots. The root was not just drinking from the soil, it was farming it, sending microorganisms out to gather resources, pulling them back in when they were full, extracting what was needed, and releasing the survivors to repeat the process. This cycle was given a formal name in the scientific literature. The researchers called it the rhizophagy cycle. The word is built from two Greek roots, meaning root and eating. Since the original 2010 paper, more than 40 follow-up studies across monocot and dicot plant species have confirmed the process. It has been observed in corn, in wheat, in sugarcane, in lettuce, in several species of native grasses, and consistently in tomato. The 2014 paper from the same Queensland team, published in Crop Science, demonstrated measurable root biomass gains in both tomato and sugarcane when living yeast was introduced to the root zone under controlled greenhouse conditions over a 10-week trial period. The practical significance of this for a home vegetable gardener is not abstract. It means that when you introduce living yeast cells to the soil around a tomato plant, you are not simply adding a nutrient solution to the water that plant passively drinks. You are stocking a hunting ground. You are putting live prey directly in the path of an active biological system that is already running continuously underground. And you are doing it for less than $1, using an organism so common and so safe that the federal government permits it in food products consumed by children. The three effects that the rhizophagy cycle research helped explain, and that the subsequent studies in 2023 and 2024 went on to quantify, work simultaneously and reinforce one another. The first is hormonal. Saccharomyces cerevisiae produces indole-3 acetic acid, abbreviated in the literature as IAA, which is the primary auxin of the plant kingdom. Auxins are the chemical signals that instruct root cells to divide, elongate, and push out new root tips. They are the master switch for lateral root development. The synthetic powders sold in garden centers for $8 to $15 per container are laboratory-produced copies of this same molecule class. The living yeast produces it naturally and delivers it directly to the root zone in combination with cytokinins, a second hormone class that accelerates cell division at the growing tip of every lateral root. The 2024 Frontiers in Industrial Microbiology paper measured significantly elevated chlorophyll levels in lettuce seedlings treated with brewer's yeast residue, and attributed the gains directly to increased auxin and cytokinin production by the living fungus in the root zone. The second effect is nutritional, and it is the one that most clearly separates a living yeast preparation from any synthetic alternative. As Saccharomyces cerevisiae multiplies in warm water during the 30-minute preparation window, the cells release amino acids, peptides, B vitamins, and a range of trace minerals, nitrogen, phosphorus, potassium, magnesium, iron, and zinc among them, into the surrounding liquid. These are not raw mineral compounds that must be broken down by soil bacteria before a root can access them. They are already in the pre-digested bioavailable forms that root hairs can absorb on direct contact. The plant does not need to spend energy processing them.
They are immediately available the moment the diluted solution reaches the root zone.
The third effect is competitive and protective. Yeast cells introduced to the root zone multiply rapidly and physically occupy space in the soil immediately surrounding the roots. Studies on lettuce and tomato noted measurably lower incidence of certain damping-off fungal pathogens in yeast-treated root zones. The mechanism appears to be a combination of simple space competition. The yeast occupies the ecological niches that pathogenic fungi would otherwise colonize, and the production of antimicrobial peptides that Saccharomyces cerevisiae secretes as a natural byproduct of its growth. These peptides have been studied in the context of food preservation for decades. Their role in protecting the root zone of a vegetable plant from soil-borne disease pressure is a newer and still developing area of the research. 3 simultaneous effects from 1 teaspoon of pantry yeast dissolved in warm water.
Hormone signal that tells roots to branch and multiply.
Nutrient delivery informs the root can use immediately. Microbial competition that reduces pathogen pressure in the root zone. And underneath all 3 of those mechanisms, the rhizophagy cycle running continuously, pulling the yeast cells inside the root tissue, extracting what they carry, releasing the survivors to gather more. The plant is doing work that no synthetic product can replicate, because the synthetic product is dead on arrival. It delivers a fixed dose of 1 or 2 molecules, and then it is finished. The living yeast keeps working as long as there's organic material in the soil for it to consume. Now, the numbers from the actual laboratory trials, because this is the place where gardening videos most commonly exaggerate and lose the trust of serious growers. And the real numbers are more than sufficient to justify every application.
The 2014 study published in Crop Science used both living and heat-killed Saccharomyces cerevisiae, applied to fertilized greenhouse soil planted with tomato and young sugarcane. Both species received the same baseline fertilization. Half the plants in each group received yeast inoculant added to the root zone. The other half received no yeast. After 10 weeks for sugarcane and 4 weeks for tomato, root biomass in the yeast-treated plants was significantly higher in both species. Nitrogen content in roots and shoots was substantially elevated. Phosphorus content followed the same pattern. The root-to-shoot ratio, the proportion of the plant's total mass that is underground, increased in both species. That last measurement matters most to a practical gardener, because it means proportionally more root system supporting the same visible plant. More engine for the same machine. A 2023 study published in the journal Plants took a different approach.
Researchers in Mexico isolated oxen-producing yeast strains from traditional maize land races, varieties of corn that have been cultivated by indigenous communities for centuries without synthetic inputs. They selected the 4 most productive strains, those producing the highest measurable concentrations of indole-3 acetic acid, and used them as soil inoculants in a controlled corn trial. The 4 strains produced, on average, a 1.5-fold increase in plant height, fresh weight, and root length compared to untreated control plants grown in identical conditions. Not synthetic chemistry, not laboratory-engineered organisms. Wild yeast strains isolated from traditional agricultural soils, doing in a cornfield what Saccharomyces cerevisiae does in a backyard tomato bed. The 2024 paper from Frontiers in Industrial Microbiology used residual brewer's yeast, the material left over after commercial beer production, applied to lettuce and tomato seedlings at a concentration of approximately 10 million cells per milliliter of solution. At that concentration, the researchers recorded measurable improvements in seed germination rate, leaf number at 3 weeks, fresh weight of the seedling at harvest, and chlorophyll content of the leaves. The chlorophyll finding is significant because chlorophyll content is a reliable proxy for the plant's ability to convert sunlight into energy. Higher chlorophyll means more photosynthetic capacity, which means more energy available for root expansion, fruit development, and stress tolerance. These are not miracle claims. They are measurements from controlled trials published in peer-reviewed journals, and they describe real, reproducible, modest but meaningful improvements in the biological metrics that determine how productive a vegetable plant becomes over the course of a full growing season. A noticeably larger root mass, higher mineral content in both roots and shoots, better germination rates, more chlorophyll. These gains compound across a full season in a way that a 4-week laboratory trial cannot fully capture. This is the gap between what the lab measures and what a backyard gardener observes by the end of August, and it is not a gap created by exaggeration. In a controlled 4-week tomato trial, root biomass gains plateau within the measurement window. The experiment ends before the plant enters the phase of growth when root expansion has its largest downstream effect on fruit size and yield. In a full 8-month backyard season with 3 strategically timed applications, the compounding effect of a larger root system, one that was established wider and deeper during the first weeks after transplant, runs forward through every subsequent stage of the plant's life. More roots in May means more water and mineral uptake in July, which means more fruit in September. The gardener pulling that plant out of the ground at the end of the season is looking at the accumulated product of that entire chain. The 2 to 3 times denser root ball that experienced home gardeners consistently report in yeast-treated plants is not an exaggeration of the laboratory data. It is the laboratory data extended across the full arc of a real growing season under real conditions, which is something a controlled greenhouse trial with a fixed 4-week window is not designed to measure. The mechanism is real. The compounding effect over time is real. The dollar cost per application is real. What remains is understanding precisely when and how to apply the preparation so that it lands at the 3 moments in the plant's life when the biological return is largest. Before that, it is worth understanding the shelf physics that have kept this preparation out of commercial distribution since the first company that might have tried to bottle it discovered the problem in an early trial. Because the story of why this cannot be sold to you is in some ways more interesting than the story of how it works. And it connects directly to why the synthetic alternatives that are sold at garden centers are, by definition, always going to deliver less than what a freshly prepared living solution can provide. The active ingredient in every commercial rooting powder, every premium root stimulator sold in a tub for $12 to $30, is a synthetic auxin, either indole 3-butyric acid, abbreviated IBA, or naphthalene acetic acid, abbreviated NAA, or in many products, a combination of both. These molecules are laboratory-produced analogs of the same hormone class that Saccharomyces cerevisiae produces naturally. They are not fraudulent products. They genuinely stimulate root initiation in cuttings and transplants, and they do so reliably and predictably, which is exactly why they dominate the retail market. A powder in a sealed tub does not require refrigeration. It does not generate gas pressure. It does not die on a warm shelf in a warehouse. It ships to any state, sits in inventory for a year, and works the same in December as it did in March. From a retail logistics perspective, it is a nearly ideal product. A living yeast preparation does none of those things. The moment Saccharomyces cerevisiae is dissolved in warm sugar water and begins to activate, it starts producing carbon dioxide as a byproduct of fermentation. At active population densities, a sealed 1-liter glass jar can accumulate enough internal gas pressure to crack within 48 hours at room temperature. A plastic bottle would bulge visibly within hours. There is no food-safe preservative or stabilizer that can suspend the organism in a dormant state without crossing the temperature or chemical threshold that kills it. Kill it, and you have a liquid containing dead cellular debris. Dead cells produce no hormones, perform no rhizophagy, and deliver no living competitive colonization to the root zone. You have spent money on a jar of biological compost tea with a laboratory label and a large price tag. The physical impossibility of shelf-stable living yeast preparation, combined with a raw ingredient cost of under $1 and 0 proprietary chemistry to protect with a patent, means there is no commercial incentive for any major garden brand to attempt what cannot be profitably solved. So the product has never appeared on a retail shelf, and it never will. The recipe, by contrast, has appeared in the notebooks of serious home gardeners in every generation since packaged dry yeast became widely available in American grocery stores after the Second World War. The science that explains why those gardeners were right did not arrive until 2010. But the observation that something was working in the root zone when yeast was present had been documented by practical experience long before a laser microscope confirmed the mechanism. The recipe has 3 ingredients and takes 30 minutes from start to finish in any kitchen using equipment every household already owns. 1 liter of non-chlorinated water, 2 tablespoons of plain dry baker's yeast, which is approximately 20 grams or 2 standard packets. 1 tablespoon of plain white sugar, nothing else. No additional nutrients, no proprietary additives, no equipment beyond a clean glass jar, a spoon, and a rubber band. The first step is temperature, and it is the step where the majority of home attempts fail without the gardener ever knowing why. Dry baker's yeast is dormant at temperatures below about 90 degrees Fahrenheit and dies rapidly above 120 degrees. The optimal activation window is between 105 and 115 degrees Fahrenheit. Both Fleischmann's and Bob's Red Mill publish this range on their packaging. Without a thermometer, the traditional field test is to press the back of the wrist against the outside of the glass jar. The water should feel warm and comfortable, but not cause any instinct to pull away. If steam is rising visibly from the surface, the water is too hot. The yeast will be dead before the sugar dissolves. If the water feels neutral or cool to the wrist, the yeast will stay dormant and the brew will sit flat with no biological activity. A reliable method for kitchens without a thermometer: bring a full kettle to a boil, pour the boiling water into the glass jar, and let the jar sit uncovered on the counter for 15 minutes. In a typical kitchen environment, the water will drop from 212 degrees into the safe activation window on its own during that time. No measuring required. The second step is dissolving. Pour the warm water into a clean glass jar with a capacity of at least 1.5 liters. Add the 2 tablespoons of yeast and the 1 tablespoon of sugar. Stir gently with a clean spoon for 30 seconds until the mixture becomes a cloudy beige liquid with no dry powder visible on the surface. The sugar is often misunderstood by first-time users. Plant roots have no biological use for refined white sugar in the soil. The sugar is not added for the plant. It is the activation signal for the yeast. Without an energy source, dry Saccharomyces cerevisiae stays in deep metabolic dormancy and produces almost no hormones, no amino acids, and no biostimulant compounds at any meaningful concentration. With sugar, it wakes, multiplies rapidly, and begins releasing the full range of compounds that drive the root response. By the time the solution is diluted and applied to the garden, the sugar has been almost entirely consumed by the yeast through fermentation. What remains in the liquid is the biological output of that fermentation: hormones, amino acids, B vitamins, and trace minerals in immediately bioavailable forms. The third step is covering. Place a single layer of clean paper towel across the opening of the jar and hold it in place with a rubber band. Do not use a tight-fitting lid under any circumstances. As the yeast population multiplies, carbon dioxide gas accumulates in the headspace of the jar at a rate significant enough to crack a sealed glass container within hours. The paper towel covering allows gas to escape continuously while keeping dust, insects, and kitchen debris out of the brew. The jar then needs to be placed in a dark location. Direct sunlight delivers ultraviolet radiation that damages yeast cell membranes and reduces hormone production. A closed pantry, a kitchen cabinet, or a shaded corner of the counter covered with a folded dish towel works correctly. 30 to 40 minutes at room temperature in darkness is the full preparation window. The visual indicators that the brew is ready are unmistakable to anyone who has ever proofed yeast for bread. A dense, foamy cap forms across the surface of the liquid. Tiny bubbles rise continuously through the body of the solution. The smell is mild, slightly yeasty, and familiar.
That foam is the visible evidence of a population of living Saccharomyces cerevisiae cells in active metabolic state, producing everything the root zone needs. When the foam appears, the concentrate is ready and must be diluted before it contacts any plant. Dilution is not optional, and the ratio is precise. 1 part brewed concentrate to 10 parts plain, non-chlorinated water. This produces approximately 11 liters of finished, ready-to-use biostimulant from the single-liter brew. Tap water left uncovered in a clean bucket for 24 hours loses most of its dissolved chlorine through natural off-gassing and works correctly. Collected rainwater is ideal. The diluted solution must be used within 24 hours of mixing. After that point, the yeast population crashes as the available food supply is exhausted. Fermentation chemistry shifts toward alcohol production, and the resulting liquid can mildly stress root tissue rather than support it. Fresh preparation each time is not optional. It is the difference between a biostimulant and a mild root stressor. Application goes directly to the soil at the base of the plant, never on foliage. The hormones and amino acids in the solution are designed to operate at the root surface. Foliar application delivers them to the wrong organ entirely and wastes the preparation. Pour slowly and evenly at the base of each plant, soaking the root zone to a depth of approximately 6 inches without creating standing water. A mature tomato or pepper plant receives approximately 1 liter of the diluted solution. A seedling or fresh transplant receives a single cup. Timing determines whether the application transforms the season or merely represents a pleasant experiment with a modest result. There are 3 moments in a vegetable plant's growing season when root growth matters enough that a well-timed yeast application produces a return measurably larger than an application made at any other point. The first is at transplant, the day a seedling moves from a starter cell into open garden soil. This is the highest stress moment in the plant's early life. Root hairs that were developed in the compressed environment of a small plastic cell are suddenly surrounded by unfamiliar soil biology, different mineral profile, different moisture distribution, and different temperature gradients. The auxin flood delivered to the root zone at this exact moment accelerates lateral root initiation and substantially reduces transplant wilt. Plants treated at transplant tend to hold their leaves firm through afternoon heat within the first week, rather than drooping and recovering slowly over 2 weeks. The second application lands at the first sign of flowering. This is the moment when the plant makes its resource allocation decision: how much energy to commit to fruit production versus continued vegetative growth. A root system that is still expanding and actively mining the surrounding soil at this stage means more flowers held to completion, fewer aborted, and better fruit set overall. The third application is timed to coincide with the appearance of the first small fruits. The plant is now entering its highest demand nutritional phase. Every calorie, every mineral molecule pulled in over the next 6 weeks goes directly into swelling fruit. A fresh wave of yeast-produced amino acids and bioavailable minerals delivered to a root system that was built wider and deeper through the first 2 applications supports that demand without the plant competing with itself for resources. 3 applications across the season. That is the complete system. More frequent applications risk shifting the soil microbial community too far in one direction. Fewer applications miss the windows where the biological return justifies the preparation time.